Hi, welcome to this webinar on food safety. I am Rejtharová, the Sales Support Director at Affinisep. Thank you very much for joining us for this webinar organized by Affinisep. Affinisep designs and manufactures polymeric sorbent for diverse applications, including solid phase extraction. Indeed, Affinisep develops and markets products for food safety, molecular biology, and environmental applications. 2 weeks ago, my colleague Luna Moles and I had the pleasure of taking part in the new International Traces conferences in Ghent. This meeting is devoted to the analysis of drug residues in food, feed, and environment within the One Health concept. This subject is of course very important, and we are pleased to organize a webinar on this field called Two Case Studies on the Monitoring of Banned Substances in Foodstuff by LC-MSMS, Beta-agonists and Nitroimidazoles. In fact, there will be two short webinars thanks to the participation of Dr. Emmanuelle Bichon from Oniris LABERCA and Dr. Martina Rejtharová from USKVBL. Dr. Emmanuelle Bichon is Head of Technical Support and Innovation at LABERCA Oniris. She will present us analytical analysis of some beta-agonists and the challenges encountered to develop the official French method. LABERCA is a joint research unit labeled by the General Directorate of Education and Research of the French Ministry of Agriculture and by INRAE. The unit studies the main classes of chemical residues and contaminants from agro-supply to food and beyond by focusing on human health effects. The unit is a national reference laboratory with the Directorate General for Food for different classes of substances. Dr. Martina Rejtharová is head of National Reference Laboratory for Residues of Banned Substances at the USKVBL, the Institute for State Control of Veterinary Biologicals and Medicines. This organization is the Veterinary Medicinal Agency of Czech Republic. Martina will present us the analysis of nitroimidazoles in diverse matrices. During the webinar, feel free to ask for any question in the chat room. Before listening to Dr. Emmanuelle Bichon, my colleague Luna is going to give a short overview of Affinisep products on sample preparation for food safety. In particular, for this webinar about banned substances, sample preparation is essential to reach the minimum method performance requirements defined by the food safety regulation.
Hi, thank you all again for coming and assisting to our webinar. So first things first, who are we? So Affinisep is a French company founded a decade ago. We are an applicated company specialized in solid phase extraction for sample preparation. We are manufacturer of all our polymeric resins. We have also an entity in the USA, and thanks to our distributor, we sell 80% of our products worldwide. So, as I told you, we are manufacturer of polymeric resins from the beginning to the end. That means that we are able to adapt our product to our customer needs in physical and also in chemical properties. So we can really create and adapt our product to your needs. Affinisep is present in 3 different fields. The main topic, and we are going to speak a bit more later about this, is in food safety. Our products are based on molecular imprinted polymer I will explain this technology. And our products are really ready to use. We sell kits for the selective extraction of some range of molecules, such as pesticides, mycotoxins, and obviously antibiotics. We are also present in environmental analysis with passive sampler and SPE disk, also for the extraction of some contaminants in environmental water, such as pesticide, drug residues. And finally, we also have products in bioanalysis and proteomics for key steps before LCMS, such as for peptide desalting, fractionation, purification. But we also have more general products. As I told you at the beginning, we are manufacturer of polymeric resins. That means that we can supply you with some more classical cartridges with L-Shelby, Sachs, wax, etc. And as we are manufacturer, our products are really reproducible and all the process is really under some control. And we have different particle size and we can adapt. We have different formats for all of our products. We have some products for automatization, like 96 well plates, but also flangeless cartridges. We have reversible cartridges and also glass cartridges if you want to avoid some plasticizer. We have LRC format, but also online and delay column. And finally, we have also disk formats, which can be useful if you want to load, for example, 1 liter in a really fast time. And for this disk and for everything, we have a large chemistry of sorbents. And we also can have some delayer if you want non-target screening. So let's be a bit more specific. As I told you, for food safety, our products are mainly based on molecularly imprinted polymer. But what it is? Well, everything is based on some template molecule. It's like the blue things, which look like your target molecule. It's not the same molecule to avoid later false positive. Well, you put this template molecule with some functional monomers, which will interact really specifically with this template molecule. It's the first step. It's called the self-assembly. Then this functional monomer will polymerize. It's the polymerization step. And after that, there is a target extraction. Thus, you will let the template go out by some washing step with some solvent. And here you have some cavity which really look like the target molecule you want to capture. Thus, when you want to load your matrices with the molecule you want, with the target molecule, it will interact really specifically with our MIP, with these cavities, and here we'll have the molecular imprint phase. So our products are a high recovery and they are really selective. And the main advantage is that you can store them at room temperature without any problem. We sell our products in kits, so it's really ready to use. Thus, you have your cartridges, but also some protocol which is really detailed and according to your need, to your matrices, and you will also have some certificate of analysis. So it's really ideal for complex matrices. It's simple, fast, ready to use, and our products are also, as I told you, able for automatization. So here are a bit overview of all of our products for food quality control. We have some families of products. So for pesticides, we have products for the analysis of glyphosate and its main metabolite AMPA. We have one cartridges which can analyze picolinic herbicides such as picloram, aminopyralid, clopyralid. But we also have products for endocrine disruptors such as all the bisphenol and estrogens. We have products for the single analysis of mycotoxins, such as AFLA, DON, fumonizids, ochratoxin, patulin, and zearalenone. But we also have products for the multi-analysis of this mycotoxin, such as if you want to analyze fumonizin with zearalenone, it's possible. And we will speak, and it's the main topic today. We have every product for antibiotics, for prohibited, but also for limited substances in foodstuff and in food contract materials, such as aminoglycoside, amphetamine, beta-agonists, etc. And we have products for pH present in oil mainly, and we have different formats if you want to analyze the 4 regulated pH and another if you want to analyze more broader pH and in more complex matrices. So now it's the time of example. So now we will speak a bit more of drug residues. So here is an example of our product for the analysis of chloramphenicol in shrimp. So chloramphenicol is an antibiotic which can cause several health for humans, even if it's present in really low concentration. That's why this molecule is prohibited, and laboratories have to detect them at really low level at 0.3 microgram per kilo. So at the left, you can see some sim chromatogram with different loading volume spiked and unspiked of shrimp with chloramphenicol. You can see that our recoveries are really high, over 95%. Obviously, we have diverse reference laboratory which have validated a protocol for concentration below this the limit of 0.3 microgram per kilo. We have a relative standard deviation between 3 and 12%. This analysis can be done by LCUV, but also by LCMS. It can be done by automatization, and obviously we have tested other matrices such as honey, milk, bovine urine. Here is the analysis of tetracycline and of 3 other epimers in meat. So tetracycline are used in livestock for therapeutics purpose, and they are effective against a broad range of bacteria, but their concentration are regulated at different level according to the matrices. For example, according to this regulation, the concentration of tetracycline in meat it's around 100 microgram per kilo, which is in liver and kidney it's around 600, and it's totally prohibited in honey. So on the left you can see some UV chromatograms of tetracycline and 3 other epimers at a concentration of 15 micrograms, spike at the middle. So you see that our recoveries are higher than 70%. We have a RSD between 2 and 12%, so it's quite low. It can be also automatized, and we have tested other matrices than meat, like honey, milk, pork, kidney, and salmon. Finally, our last example is about aminoglycosides in foodstuff. So aminoglycosides are also used to treat severe infection in animals, and our concentration are regulating in Europe at different level according to the aminoglycoside and according to the matrices. So here you can see some example of aminoglycoside with the MRL level. So thanks to Nestle, they have supervised and carried out all the AOAC method, which have been validated now and can be useful. You can find our poster on our website if you want more information. So they found a really low relative standard deviation, and they have tested a lot of matrices like honey, milk, bovine urine, infant formula, etc. Just to conclude, let's speak a bit about our product for proteomics. So our product can be used for allergen detection, to check the adulteration, but also to verify the origin of animal products. And our products are in 2 different ranges. The first is called Attract SPE, and it's based on chemistry. That means that you can tell us if you want some sorbents on silica-based or polymeric retines. So like you can see all of our different sorbents available. And on the other hand, the second range called BioSPE is more ready to use. It's when you want when you have a specific application and you want to use it. So for example, if you want to use it for peptide desalting, fractionation, purification, we can provide you some really ready to use kits. And all of these products in the 2 different ranges are available at different formats. So you can have some tips, but also plates if you want to go to automatization, 96 and also 384 well plates. But you also have some spin columns and obviously SP cartridges. So, well, as I told you at the beginning, 80% of our customers are worldwide, and we thanks them to to trust us. And I wanted to thank you all for listening of this brief introduction of Affinisep.
Thank you very much, Luna. Now we will follow the discussion with Dr. Emmanuelle Bichon, Head of Technical Support and Innovation at LABERCA. She will present us analytical analysis of some beta-agonists and the challenge encountered to develop the official French method.
Thank you, Michel, for this kind introduction. I just share my screen. Okay. Okay, so I will present you what we can do for 15 years now on molecular imprinted polymers. And this application today is dedicated to beta-agonist monitoring in feed sample. And we are in charge of that development. Perhaps I can put this part up. Because we are the national reference laboratory in France for growth promoting agent in use in feed sample, and we are in charge of development of this method to control misuse of this growth promoting agent. And according to the regulation 2022-1644, beta-agonists are included in the list of prohibited or unauthorized pharmacologically active substances in its Annex 1. And it doesn't work. Oops. Okay. And in this Annex 1, yes, you can see that beta-agonists are a substance with hormonal and thyrostatic action, and beta-agonists for which it is prohibited to use under the Council Directive 96/22. So we have to develop strategies, analytical strategies to face the use of this compound, particularly in feed samples. And we published in 2018 a protocol to analyze several beta-agonists so there are only 10 here, but we are much more in the method with my colleague Loïc Herpin, where we develop a strategy for controlling either polar and apolar compound using a USFC MSMS approach. So this, but before when we were before 2012, we first analyzed this compound from feed by this sample prep, where we did first liquid solid extraction with methanol and acetate buffer, and after shaking and centrifuging the extracts, we evaporate the supernatant and centrifuge before to combine two different SPE. The first one was a C18 SPE, and after cation exchange SPE. And then the extracts were reconstituted in a mixture of methanol and water before analysis in LC or SFC. And we use both detection modes, so analysis mode, so first MSMS, traditional transition mode, but also neutral loss, focused on the water loss in order to screen under suspect screening new beta-agonists. What we've seen at this stage, it was a loss of the most polar beta-agonists, and because the cationic exchanger was not enough retentive for the most polar compounds. So we decided to change a little bit, and the C18 was not adapted to retain this polar compound. So we adapt filtration before cationic exchange. It was not so bad, but we observed low quality of the extract and also very complex chromatogram coming from the neutral loss acquisition mode. So we decided to work on other new support, which was manufactured by a Sweden company, and we were able to purify the specifically beta-agonists. So as Luna said, it's molecular imprinted polymers, and we really increase the extraction quality with very cleaner extracts. And for the suspect screening approach, we obtained a very easier chromatogram to analyze the neutral loss of water for these chromatograms, from these chromatograms. So as you can see, I give you an example here of 6 of the beta-agonists of interest. So salbutamol, zilbaterol, bromobuterol, isoxsuprine, ractopamine, which are isomers, and clenbuterol. Here in the blank sample, and here when we spiked at 50 microgram per gram, the feed sample. So it was very efficient for the detection of this compound. But in 2019, the first MIP generation was very difficult to purchase because of provider difficulties, I think, and it was not so commercially available. So we decided to contact Affinisep Society because we worked a lot with them for their application, particularly bisphenol and estrogens, MIP. And they we proposed together to develop these similar polymers, as we have already, but we have another needs is to try to have a better percolation flow rate, because it was sometimes very long to pass the sample through the column. And Affinisep proposed in 2020 a new support, so it's a MIP dedicated to beta-agonists, and where we adapt the method. So here you can see a feed sample. So we add in this feed sample only 0.5 ppb of beta-agonists, and we also include the deuterated standards for the internal standards. And with the same extraction and just a liquid extraction before filtration to help the filtration to go faster, we put the new MIP, and we managed to increase a little bit more the selectivity from the first generation of MIP to the second one. Here you have the example of a beta-agonist we managed to detect very well now with this new generation from Affinisep, comparing to the first generation here. So we then decided to validate this method for all the beta-agonists we monitored in the lab at a level of 0.25 ppb. And today we are able to propose in the official French method the MIP solution for the beta-agonist control. And we achieved also the validation according to the new, it's not so new, but for us it's a new regulation 2021-808 to monitor growth promoting agent, but in general pharmacological substances. And at the end, we also managed to accreditate the method according to the ISO 17025 2017 standards. So I'm happy to share that work with you, and it's the work of my colleagues Johan de Seninck and Loïc Herpin. So I would like to honor them very warmly, and all the team of the LABERCA for this work also. And thank you for your attention. If you have any question after the presentation of Martina, I will be happy to answer.
Thanks a lot, Dr. Emmanuelle Bichon, for this very nice presentation. Now we will listen to Dr. Martina Rejtharová, Head of National Reference Laboratory for Residues of Banned Substance at the USKVBL, the Institute for State Control of Veterinary Biologicals and Medicines. She will explain the determination of 11 nitroimidazoles in biological matrices by LC-MS using AFFINIMIP SPE for sample preparation. Thanks a lot, Martina.
Okay, thank you, Michelle, for your introduction. And I will also try to share my screen. Okay, thank you so much. As Michelle said, I will speak about determination of nitroimidazoles in biological matrices using AFFINIMIP SPE for sample preparation. Before I would like to say that I work in the institute, which is the Veterinary Medicinal Agency for Czech Republic as the official authority, and we are also responsible for food safety in this field. I come from National Reference Laboratory for Residues of Banned Substances, preferably in food of animal origin. As Emmanuelle explained, we work under the scope of the European Regulation 1644 from 2022 under the residue official controls. And our laboratory is responsible for hormonally active substances and banned medicines. For these substances, we use GCMS and LCMS methods. And we apply that methods for various matrices, which include samples from live animals like urine, or blood, or hair, or feathers, and also from slaughtered animals, which are tissues like muscle, liver, kidney fat, and others. We also analyze products like milk, eggs, and honey, and of course also feeding stuffs and feeding water. And similarly to LABERCA, all our methods are included under the scope of accreditation for our laboratory according to ISO standard 17025. And of course, all of them are fully validated according to this new validation, new regulation. And this validation is according to regulation of the European Union 808 from 2021. Now, speaking about nitroimidazoles, they are pharmaceutically active substances, and all of them are fully prohibited for food-producing animals. Here you can see the list of nitroimidazoles. First three of them, metronidazole, dimetridazole, and ronidazole, are especially prohibited because they are included in Table 2 of Regulation 37 from 2010. But the other nitroimidazoles are also prohibited for food-producing animals because they are not included in Table 1 of this regulation, European Regulation 37 from 2010. That means that they cannot be used for food-producing animals. Our method covers both parent substances and also some metabolites. And I would like to mention carnidazole, which is here in orange color, and also you can see the structure of this substance. Because carnidazole has slightly different chemical properties than the other molecules of nitroimidazoles, and it has some influence to the determination, which I will describe later. In our laboratory, we applied 2 different cleaning protocols for sample preparation before LC-MS measurement. The first procedure is based on a method which was obtained from European Reference Laboratory for nitroimidazoles, which is BVL in Berlin. And this extraction method is based on acetonitril extraction and then on extraction with ethyl acetate from phosphate buffer. Another protocol for sample preparation is based on AFFINIMIP cartridges. Prior to MIP procedure, we also apply this acetonitril extraction, and then we apply the protocol which is recommended by the producer of the MIP column. This protocol consists of conditioning, of course, and then loading sample, some washing steps, and elution. Altogether, it's quite simple protocol, easy to apply. Conditioning is done with toluene, acetonitrile, and ammonium acetate. Then the sample extract is loaded, and some washing steps are done with water and some organic solvents. And finally, the elution is carried with 60% of acetonitrile with some acetic acid. Our method covers also internal standards. We apply 9 isotopically labeled internal standards related to the substances which are determined. For measurement, we use HPLC Agilent coupled with triple quadruple MS detector. And the concentrations which we apply are from 1 to 10 ppb for all matrices. I would like to mention 3 critical points which are important for nitroimidazole determination. The first comment is about adsorption on glass surfaces. We know that some nitroimidazoles easily adsorb on glass surfaces, so it is recommended to use polypropylene tubes and avoid any glass surfaces. The second comment comes to the flow rate when we use MIP columns, because it is important to keep very low flow rate for especially sample loading and sample elution, because only this slow flow rate may ensure the high recovery from the columns, from the cartridges. And the last comment comes to evaporation, because for some substances within the nitroimidazoles, especially for metabolites, it is important to gently evaporate the extracts under the nitrogen stream and finish before the complete dryness, because the too intensive drying may result in the loss of the substances. Now I would like to compare these 2 extraction protocols which we applied. I may say that both of these procedures provide clean extracts for LC-MS measurement. And what's important, the sufficient sensitivity for all tested matrices. And what does it mean, sufficient sensitivity? As was mentioned before, that means that CC alpha and CC beta, according to 808 European regulation from 2021, these decision limits and detection capabilities are lower than minimum method performance requirements, which are set by European Reference Laboratory for nitroimidazoles at 1 ppb for all matrices. So that means that with this method, we can reach the CC alpha and CC beta lower than 1 ppb. Now I come again to the list of substances which we analyze within our method, and I bring your attention again to the carnidazole, because here you can see the results of 2 applied cleaning protocols. With the liquid-liquid extraction, we could validate 11 analytes And unfortunately, the carnidazole was not present in the extract because, as I said, carnidazole has slightly different chemical properties. So, and it is not possible to extract it with this applied extraction method. With the MIP procedure with AFFINIMIP SPE, we were able to analyze carnidazole. That means that we could validate all 12 substances within this method, and carnidazole was present in all matrices. Here I would like to share with you a few results from our validation parameters. Of course, we observed the linearity for all matrices, and we realized that for all matrices and all substances, the response is very linear. That means that the correlation coefficient R squared is higher than 0.99. The first example is about ronidazole in milk, and you see that the linearity is very good. Another example is dimetridazole in poultry muscle. Also, the linearity was quite nice. Here is the metabolite of dimetridazole in eggs, and also in eggs, the linearity was working well. And finally, carnidazole in plasma, also well working and linear. Another validation parameters are regarding the sensitivity. As I said before, CC alpha are lower than 1 ppb. You can see all the substances, including metabolites, in 5 matrices: eggs, plasma, milk, muscle, and honey. And all these CC alpha, these decision limits are lower than 1. Some substances are better, for example, ipronidazole or hydroxyipronidazole. The CC alpha are at 0.5. For some substances, the CC alpha are slightly higher. For example, metronidazole has low response, so it results into the CC alpha at 0.9. Here, I would like to show you a few chromatograms. These are chromatograms of nitroimidazoles spiked into bovine milk. And this spiking was done at the level 1 ppb, that means at the level of MMPR. And you see that these chromatograms are quite clear, clean from any impurities, and we can easily see peaks with good signal-to-noise ratios. Here are the purest chromatograms, but when I say purest, that means that the peaks are not so high, but still very well visible. Hydroxymetronidazole in black line, you can see that still at level 1 ppb in bovine milk is still well visible. And the last group of substances included carnidazole in green line, also nice peaks, nice chromatograms, also at a very low level in bovine milk. To conclude, I must say that the title of my presentation was not correct because I spoke about 11 nitroimidazoles, but this is not true. We may say that with this method, we are able to determine 12 nitroimidazoles, and it was some improvement of our method. So thank you to AFFINIMIP for that columns. And of course, I would like to thank my colleagues who develop and provide this method. And I thank you all for your attention. And the last reference comes to one of our residue study, which we published a few years ago. And we observed the residues of nitroimidazoles in chickens in various matrices. And I would like to say that for all these samples, we also used MIP columns. So this is very nice and applicable method. So thank you again for your attention.
Thanks a lot to Martina for this very clear explanation for all the results. I remind that you can write your question on the chat room. Martina, I have one question. So we understand that instead of 11, you could analyze 12 nitroimidazoles. Are there some matrices more complex to be analyzed in your case?
You mean that there are some other possible to?
Yes, matrices like is a meat more complex than Oni or?
Yes, we tried different matrices, including feed, for example, which is also very hard matrix, and yes, we are able to try a new matrices and also new substances which can be included in this method. And we were surprised that this is applicable for such a wide scope of analytes and matrices, because we knew that the MIP columns are very specific, but still it is possible to apply it to various substances within this group.
Thanks a lot. There are some questions for Emmanuelle from Florent. Do you have a list of beta-agonists? We know that you have shown some results of about 5 or 6 beta-agonists. Do you have a list of beta-agonists retained on the MIP, or is it confidential, or you cannot supply it, maybe?
Yes, so it's totally true. We have a list, but it's a confidential list because of the control of forbidden substances. We are obliged to keep close this list and totally, blind in order to ensure an efficient control. But I can I can say something that all the beta-agonists tested on the MIP, we are several dozen of beta-agonists, are able to be retained of MIP. Not all at very high recovery yield, but as we are controlling the presence of the beta-agonist, it's not necessary to reach very high recovery yield, because trueness is not the main goal. The presence of this compound in feed is the result we want to control. So until now, all the beta-agonists tested on the MIP present a certain retention, in general very high retention on MIP and high selectivity. So it's very nice because we compare the result. I don't present it here, but on the immunoaffinity chromatographic approach, and in this case we have some loss of certain beta-agonists because yes, we try to mix antibodies, but it's not so efficient, and it's also difficult to store and to use extra. So MIP is a very good strategy to enlarge and to imagine other retention of other beta-agonists on this list.
Thanks a lot. There is several questions from Gershom. One of them is about beta-agonists. How often does the beta-agonist analyzed in a year? In France, for instance.
Do you mean how many samples?
I don't know.
Because it's yes, the samples analyzed in the national control plant are totally planned by the authority. So we have several hundred of samples analyzed for feed, for example. But and I think in Europe it's much more. Because it's a percentage of animal production control. So, and each state has also to control a certain production. So it depends on countries. But in France, I think on feed sample we are around 300 or 400 samples per year for the official control. But I think there is also quality control plan for private industry. I just know the public control, official control.
Okay. There is a question. I don't know if one of you can answer this question. What are the maximum residue levels of pharmaceuticals in food crop? Do you have any information about that? No, I think that
it's I can begin a few answers, and Martina, if you want to add something, I will be happy to yes have additional information. But there are perhaps two ways to answer that. The first one is if they are forbidden, so yes, there is no limit. There's only the presence of the compound is forbidden. So, and as Martina said, we have in this case minimal method performance requirements. So we have to reach a certain low level of analytical performance to be reached to be sure that the control is efficient. And on the other side, if the pharmacologically substances is authorized, we have regulatory limits which are described in the document regulation 2010 37, and you have a level per feed or food sample authorized for these authorized substances. Martina, I don't know if you want to complete.
Yes, this is fully true, and this is nothing to add. So for prohibited substances, there is no limit, and for authorized substances, there are some regulation for cross-contamination cases, but then MMPRs, but no MRLs.
Okay. There was another question of Gershom. If the MIP can be applied for multi-residue analysis. Usually when we develop MIPs, molecularly imprinted polymer, we try to develop for a family of compounds like bisphenols to try to catch all the bisphenols or like beta-agonists, etc. There is few cases we have multi-residue like multi-mycotoxins, but in most cases we try to target a family of compound to keep some selectivity and to have efficient washing steps. I don't think there is more questions. So thanks a lot, Martina and Emmanuelle, for this very nice presentation and webinar. Thanks a lot for all attendees. Thanks a lot, everyone.
Thank you and bye.
Thank you. Bye.